Good afternoon, everybody at both sides of the screen. I'm Danis Kriens, Director of the Research Institute at Valsts Clinical University Hospital, and sincerely welcome you to this very important research forum. So, at the time when consequences of the COVID-19 pandemic are being tackled, the health sector and medical research are under particular pressure, and medical research in the shadow of the pandemic is particularly challenging. So new challenges cannot be solved with old methods. So innovation and new technologies are needed. So in other words, medical research has now become a necessity, and innovations in medicine and medical technology are not only desirable, but critically needed. So in this context, I'm proud that our institution has been part of such development. Since last year, the hospital's research institution scientists, under the exceptional leadership of Professor Valsts had been worked intensively developing the state research programs technology project 3T. So we are proud of the work done by our researchers and the results achieved by this project. So let me start today's forum with a short video about our hospital's research institution, under which this ambitious project has been developed. Please, video.
Latvia is located in Northern Europe, on the eastern shore of the Baltic Sea. Our country is rich in rivers, lakes, and plains. Through music, art, culture, Latvian masters have enchanted viewers and listeners all over the world. Modern solutions in electronics, information technology. Yes, we did not invent the internet, but we have the fastest internet in the world. The Research Institute of Project Stradini's Clinical University Hospital has been operating since June 1st, 2015. Education and science. These are the cornerstones and core values of our business. Our mission? To be among the leading clinical research centers in Northern Europe, discover creative and innovative findings in medical and health sciences, improve public health and promote quality of life, as well as contribute to the competitiveness of Europe. Our goal? To ensure the development of health and medical science by conducting fundamental and industrial research, experimental developments, and knowledge transfer. We contribute to the growth of future Latvian healthcare specialists. The best of the best improve their knowledge, experience, and competences with us. Versatile and comprehensive postgraduate education programs ensure continuous professional improvement of Latvian healthcare experts. The Research Institute has established the only specialized clinical research center in the Baltics with the necessary facilities and personnel for conducting studies. The center operates in strict accordance with the guidelines of good clinical practice. We cooperate with major actors in the pharmaceutical and clinical research industry. The hospital simultaneously conducts more than 150 clinical trials regarding medicinal products and medical devices intended for human use. More than 40 researchers are involved and dozens of employees are working under their lead. Clinical trials are carried out over a vast range of medical fields. Particular emphasis is put on cardiology, ophthalmology, gastroenterology and digestive disorders, endocrinology, and metabolic diseases, cardiac and vascular surgery, neurology, especially neurodegenerative disorders, and oncology. Our partners highly appreciate the quality of our research and patient engagement capacity. Our research is performed with high quality and reliability verified by international audits. The research done in the hospital is regularly audited by inspectors from both the US FDA and the European Medicines Agency. The Research Institute consists of 70 elected researchers who have produced excellent scientific publications and have participated in the most important professional fora in the field. Our scientists are opinion leaders in the global scientific and professional community. We participate in the development of European and global clinical guidelines and algorithms. A reliable and internationally acclaimed partner in scientific research.
So we have pleasure, we received the congratulations and greetings from the Minister of Health, Mr. Daniels Pauļuts. Please, his video, please.
Ladies and gentlemen, medical professionals, researchers, it is a pleasure to welcome you at this conference today. I welcome you from my home. We ask our people to work from home as much as they possibly can, so I do the same. This is a time of crisis still, but it's also a time of opportunity for advancing medicine, science, and innovation. I salute performance and capabilities of Latvian medical profession, as well as that of colleagues from elsewhere, in responding to challenges posed by COVID-19. We are managing quite well. Yet work of our countries, researchers and scientists, in finding tools and new opportunities to help patients and society as a whole in their fight against the consequences and effects of the virus should also be pointed out, appreciated, and promoted. Latvia, similarly to other countries, has allocated funding for COVID research. Scientists have had some 5 million euros available so far for various projects, as well as additional funds which have been redistributed from European funding. First results have been reported and some of research outcomes implemented in practice already. Today, 3 unique product prototypes will be presented to you at this conference, which can help mitigate impact of the pandemic in real life. First, rapid saliva tests. Second, a special device to monitor the epidemiological safety in confined space. And last but not least, a device to regulate oxygen flow for patients with COVID-19 related pneumonia. We need to find ways to bring these and other achievements of our scientists into real life without undue delay, so that they can serve their intended purpose and help us all come back to a more normal life as soon as possible. I thank you for your endeavors. I wish you every luck in your important work and a productive day.
Thank you very much for kind words. And now I would like to give the floor to the leader and father of the 3T project, Professor Valsts Pirāgs, and hopefully he will explain what does it mean, this 3T, which is very intriguing name of the project. Valsts?
Yeah, thank you, Professor Krieviņš, and thank you for this nice introduction about the Research Institute of Valsts Clinical University Hospital. And I am also part of the hospital team. I'm a clinician, but I have a very long history of research, starting from Germany, where I was defending my PhD thesis, and now I'm back in Latvia for more than 20 years, and really life here is quite exciting and rapidly changing, and the COVID-19 pandemic was not so actually challenging for Latvia, because we have challenges every year. 3T, as you asked me what 3T project means. 3T means abbreviation from tracing, testing and treatment of COVID-19 in this case. And we decided to participate in this competition, international competition, announced by the Latvian government. The pretenders were very scrutinally evaluated by external international panels of renowned researchers. So we were lucky to have this project, and if Minister said that there was a €5 million funding, actually we were just one project out of 10, so we got slightly less than €500, but it was quite challenging to keep our promises to develop at least 3 advanced technologies in very short time span, just 3 months, plus 3 months, so 6 months. I was just a captain of the young, relatively young, enthusiastic team, comprised from the researchers from our research institute, of our hospital, then from the University of Latvia, mainly from the Institute of Computing Technologies and the Faculty of Computing. So, and the third partner was, I think, the best biomedical research institute in Latvia. It is Latvian Biomedical Research and Study Center. So with this enthusiastic team, we really achieved what was promised. We developed 3 very unique technologies, and I will give a floor one by one to the leaders of those 3 sub-projects of our project. So, and I would like to start with Dr. Jānis Virbulis from the University of Latvia. Please, a short video.
Mūsu institūta izstrādes mērķis bija izveidot tādu automatizētu sistēmu, kas bez cilvēku roka darba nodrošinātu iespēju ilgtermiņā monitorēt riskus, kas darbiniekiem, studentiem, skolniekiem rodas uzturotos vai strādājot telpās, un tādējādi arī aizsargāt viņus pret iespējamo infekciju, šī gadījumā Covid, bet nākotnē varbūt pret citām līdzīgām infekcijām. Šī gadījumā nācās apvienot gan elektroniķu, gan arī programmētāju prasmes un spēkus, lai šādu sistēmu izveidotu, un sistēmas īpatnība ir tā, ka viņu var ērti izveidot arī kā papildinājumu jau esošajām gudro ēku vadības sistēmām. Mūsdienu ēkās tādas sistēmas jau faktiski darbojās.
Skaitliski aprēķini palīdz mums labāk saprast, kāda ir gaisa plūsma telpā. Aprēķinā mēs varam viegli izmainīt sistēmas parametrus, piemēram, ventilācijas apjomu, un redzēt, kādu ietekmi tas atstāja, piemēram, uz gaisa plūsmas, ātrumu sadalījumu un ogliskāvās gāzes koncentrāciju.
Good afternoon. So I think I can start already. Thank you for the nice introduction. I will present, my name is Jānis Virbulis, and I'm from Institute of Numerical Modeling of University of Latvia, as my colleagues also you see here. And I will present one development of this work package 1. And this is a model and measurement system for prediction of indoor COVID-19 infection risk. Please, next slide. Okay, after short introduction, I will first tell about the model, how we build it, and after that about the measurement system which is needed for the model, and at the end results in some basic scenarios where we can show what the model alone can show us without the coupling with this measurement system. And at the end, I will demonstrate some results in real rooms what we have seen. Next, please. So the scientific community in last time already started to believe that we will have to live together with this new virus for some longer time. Yeah. And yeah, there are mainly three routes of virus transport. The first is a direct transport of droplets from infected person to another person. The second is through the contact with surfaces, and the third is via aerosols. So whilst the first two can be considerably eliminated, holding 2-meter distance and wearing masks and washing hands, yeah, the last route, this aerosol transmission, remains therefore as a main factor in at least at indoor environments. And therefore we built this system of COVID-19 risk prediction, which consists of a mathematical and numerical model for virus transport in room and prediction of infection risk. And the second part is a measurement system, which is needed to get these parameters for the risk prediction. Next slide, please. So here we see the schematics of the model. The model is actually integral and parametric or zero-dimensional, and it handles only or mainly the mean values in the room. So in the room we have some source of virus. It happens through expulsion by speaking, breathing, or sneezing. Then further the droplets in the air are sedimenting first, the process 3, due to gravity, but also they are very fast evaporating and reducing their size, and at the end the size gets so small. In our case, we assume 5 microns is a size where we can assume that these droplets will stay forever in the air. Practically, the gravity is not more working on so small particles. Of course, the ventilation, the process sphere will transport out this aerosol. There is also some absorption of aerosol particles on all surfaces, and also there is a reduction of viability of virus when it is in the form of aerosol. And the last and not least, the process 7. These aerosol and particles and the droplets are inhaled by a person under risk, and so the risk of infection is increasing then. Yeah, we have several parameters in the room: the room size, the volume, the surface areas. We know the temperature, the humidity, and CO2 concentration in the room, and calculate also then the number of aerosol particles and number of virions and number of droplets in the air. Yeah, and yeah, and then we use this here shown processes, physical processes, to parameter. They are all parameterized in this and put together in one numerical model, which at the end predicts the risk of infection. Please, next slide. On the next slide here we see practically the sources from which literature we took the particular models, and also the model parameters are here also shown, but I don't have time to explain it in detail. I will only say that the last 7 points, the inhalation model, so we count how many virions are absorbed by a person, and from this we calculate the risk. There are this formula for R, and also it means that we, for example, when 300 virions are absorbed, it means the infection risk of 50%. Next slide, please. Okay, so on the left side we have the parameters which are needed by the model, and on the right side we have here the sensors, yeah, which we use for the measurement of these parameters. And it's simple, of course. Temperature measurement means temperature in model, the same for humidity. And then we have image sensor, which for us gives the number of people in the room. Then we have sound sensor, which recognizes sources, this expel of virus by potentially ill person as speech or sneezing. And then we have CO2 sensor, which measures indirectly measures the ventilation intensity. And we tested also particle sensors, but found that it is hard to distinguish between aerosols, which are produced by particles, expelled particles, and just dust in the room. So it's not used in the actual system. Next slide, please. Okay, and here just two examples of most complicated sensors. We use artificial intelligence here for recognition of persons and of this sneezing or coughing events. So on the left side example, we have image. Then we send this image to our computer, high-performance computer cluster, and with trained neural network, we recognize the number of persons in the room. On the right side, the same, similar happens with the sound. So the sound is transferred to spectrum, and this spectrum is sent to neural network, and the network recognizes, in this case, in this example, that this sound was sneezing with a probability of 70%, and then we accept it, this is sneezing. Next slide, please. Yeah, and the protection of privacy, of course, is also a point, but yeah, it is. We are working on this also. So here's a more physical schematics of our system. So practically we have a box or system in a room or in many rooms. On the right side, where we have sensor system and some risk visualization system. On the left side, we have our high-performance computer cluster in our institute, where we run virtual servers for this model and for neural networks of sound and image recognition. And we have in the middle also in the cloud our colleagues from Faculty of Computer Science, University of Latvia, built a cloud server which transfers the data between these two boxes, I would say. So we have sensors here. The measurements are done in the room. They are sent to server, and then the model is working and gives back the risk to measurement system or to building management system or to any other systems which want to read this data. Next slide, please. So that's all about the model. And now some results. So basic results, just with, we can learn a lot just playing with the model alone. And here we have such a situation, just took a room 3 × 3 × 3 meters with no ventilation at the beginning, with no contamination. That means there is no aerosol and viruses in the room. And 2 persons, one infected and one healthy person, are entering the room. And what happens in next 15 minutes with the risk of infection for healthy person is shown in these 2 graphs. On the left side, in logarithmic scale, we see how different scenarios what the infected person is making influences the risk. So the breathing is relatively low risk. Speaking increases the risk by an order, means 10 times. The coughing increases further the risk. The coughing once per minute and sneezing per minute increases the risk considerably. We see here also after 15 minutes, we have already a 10% infection risk in this room. On the right side, there is demonstrations that, of course, different infected persons have different concentration of virus in these expelled droplets, yeah. And when average infected person has this risk of from average infected person, we have infection risks after 15 minutes about 10%. Yeah, I would assume that this corresponds approximately to this close contact definition, 15 minutes and 2 meter distance. Yeah, we have in our case now risk from 10%. But when we meet some person with high virus concentration, then already you see after 5 minutes the risk of infection is almost 100%. So it's inevitable, but small possibilities that you will get infected in such conditions. Next slide, please. So very interesting is to understand what happens with these virions when they are expelled. Yeah, on the left side we have the same, only for 30 seconds that the infected person is breathing. Yeah, and in room with different relative humidities also here are shown, and we see that the number of virions in air is increasing all the time, but a lot of them are also sedimented on the surfaces. Approximately 50% are sedimented already, and only a small percent is going over to aerosol. It means, and also on the right side in graph you see that also for longer times, 3 minutes or longer, the virion concentration in air is mainly in these large droplets, and the droplets are not sedimenting so fast. They are mixed with the ventilation, with airflow in the air and holding there. It is, so what I want to say here, that the main virions are living in large droplets which can be good filtered by masks, for example, and I think therefore also this wearing of masks is so important, but also our model can show here. Next slide, please. So the last simple result is the influence of ventilation. It's of course one of what we can control good. Yes, we can open doors, we can open windows. Yeah, and here you see how the risk of infection is growing during 1 hour for different ventilations. And what is interesting here, that it is so that we need a very intensive ventilation to reduce the infection risk. So with the normal ventilation, so-called, as in prescribed by Latvian building code, it's būvnormatīvs, we have practically no reduction or very small reduction of infection risk, and we really need practically to open the windows to reduce the infection risk for several times. Next slide, please. So, and now real results in operational environment in one of our office rooms in university. On the left side, you see during 2 days how the number of people is changing in this room and how the CO2 concentration measured blue curve is changing, and our CO2 concentration in model. Yeah, here we see that the ventilation probably is switched on and off during the night. Yeah, and our model do not know this, that the ventilation is switched off. Yeah, therefore there is some disagreement, but we will improve this in the next slide. And on the right side, practically this most important result is we see the number of people here during 2 days. We see the speaking events, coughing events, and one sneezing event. Yeah, and we see at the end with the green curves how the infection risk is increasing. In this case, the risk is approximately 0.2%. It means the risk is low. People are just they are working and breathing there and speaking, not so much scientists here in university. Okay, next slide, please. And here we introduced one algorithm that we using the measured CO2 concentration and the number of people in the room, we can adapt and calculate the unknown ventilation intensity, which is very important because all the time the doors are open, the windows are open and closed, so we really don't know the ventilation, and these two sensors help us to calculate this. So, and on the right side again, the infection risk is only slowly decreased in this case when the ventilation was more properly calculated. Okay, next slide. And this is actually the last result. And here I compare two rooms. On the left, the room I already told about in our university. On the right side is a doctor's room in Stradini Clinical University Hospital, room 101. Yeah, and we see on the in the hospital there is not so good ventilation. Maybe there are more people in the room. They are more speaking, and also sometimes more sneezing, and therefore, as a logical result, we see that the infection risk is approximately 2 times higher in these rooms than in the room we considered before. But anyway, that is not very high. I would say that's okay. So next slide, please. And here a bit outlook of our results. So we unexpectedly reached the technology readiness level 7 already. It was not planned that we will go so far in this project. So it means we have a system prototype demonstration in operational environment. It means the system is working. And we can propose already some possible application design. On the right side, we have a box in the room or in rooms. Yeah, of course, not one room, but in many rooms, which communicate with wireless protocol to internet. On the left side, we have some cloud server and for the model and this image and sound recognition. And we see that we need for 100 systems, we need approximately 1 server. Yeah, and when the whole system would cost under 200 euro, we so expect maybe only 100, then this server part for 1 system will cost then maybe 20 euros. Okay, and of course, all kinds of notifications are possible also in this system on mobile phone or special system. So next slide, please. And this is already the last slide. I don't know how much time I have, probably. Okay, then shortly, we developed this model together with measurement system, which can predict infection risk in real time and reach the TRL7. So we can increase the safety in the room, and the parameters study shows that infection risk slightly increases for lower humidity and decreases with very intensive ventilation. And the coughing and especially sneezing events strongly increase the risk in infection and should be it's absolutely necessary to measure the presence of such events. And at the end also, we developed a method how to determine the ventilation intensity from this sensor system. Thank you very much.
So thank you very much, Drs. Virbulis and the whole team of mathematicians from the University of Latvia Institute of Numerical Computing. That is the official name. And I think this is amazing how fast you really developed this model and the first prototype on TRL7 level. So the next team I want to present will be led by Professor Renate Ranka, and this team is consists of molecular biologists from the Latvian Biomedical Center of Research and Studies. So the floor is yours.
Good afternoon, dear audience, and I would like to present our results, which are achieved in this 3T project in work package 2, which was related to testing technologies. And I can just easily to tell that it was like evaluation of novel antigen-based rapid detection test for the diagnosis of SARS-CoV-2 virus in saliva samples. Next slide, please. So a little bit about testing and about this COVID-19. So testing is a part of the first-line defense against this infection because it enables early identification and isolation of cases, and we really need it to slow transmission and to provide this targeted care to those affected and also to protect all health system operations. And this huge surge of COVID-19 cases increased demand for testing and also it put a high pressure on laboratory capacities. And tests which are used in surveillance regimes intended to reduce this population prevalence of a virus, they need to return results really quickly because we need to limit this asymptomatic spread. And we would like that these tests could be sufficiently inexpensive and also easy to execute because we need to allow this frequent testing. And so at this moment, this development and validation of such rapid and easy to use diagnostic methods is a very high priority. Next slide, please. So when we are talking about this COVID testing, different types of this COVID tests exist. And this performance depends on this test and what we need to achieve. So basically, there are three different these tests, and the probability of a positive result, it varies depending what test is used and when. So most often we are using these PCR-based tests, and on this graph it's represented by gray line, and these tests can detect really small amounts of viral genetic material, and so it's a big plus, but also this test can be positive long after a person stops being infectious because it's really sensitive test. But also this test is quite labor demanding, so we need special laboratories, special materials to perform these kits. A second test, it's called rapid antigen tests, and these tests can detect the presence of viral proteins. It's why it's called antigen tests. And so the most positive moment is that these tests can return these positive results in a very short time. And also this test is most effective when the person is most infectious. So it's like the onset of infection, approximately in the first week of infection. And also antibody tests are available, and these antibody tests detect the body's immune response to the virus. These tests are not effective at the earliest phase of infection, but it's really important sometimes to monitor these antibody levels in persons. Next slide, please. So about this testing, I must say that no test gives a 100% accurate result. And a systematic review of the accuracy of this, the most widely used RT-PCR method reported that false negative results, and these rates are between 2 and 29%. So the sensitivity of this test is approximately 71 to 98%. And this also accuracy of the viral RNA swabs in clinical practice. This accuracy varies depending on site and quality of sampling, as well as on stage of the disease and also the viral load. And on this slide, in this blue box, you can see this result from only one study, which was conducted in hospitalized patients. And these PCR test results, positive results, depends on sample time. For example, these lavage fluid specimens were most positive, 93% were positive this test. In sputum in sputum samples, 72% were positive, and only 63% of nasal swabs were positive, and feces and blood was even less. So it's like one example that this quality of sample and what sample we are using is very important. And very similarly, the accuracy of this rapid antigen test depends on many different factors, including this time from onset of infection, the viral load, so how many viral materials in the sample, the quality and processing of specimen collected from a person, how the specimen is collected, how fast it's processed, and also the precise formulation of the kit itself. Next slide, please. And so these PCR-based assays, like these are standard methods for detecting this virus right now. And these tests primarily use this nasopharyngeal swabs samples. But unfortunately, this collection of swabs is not very comfortable for patients. And also, this collection poses certain challenges, including this exposure risk to healthcare workers. And also, their reports the self-collection of such samples is very difficult and less sensitive. Thus, during the last months, several studies turned this attention to saliva samples, and some results show that indeed these saliva samples could provide high sensitivity and specificity. Therefore, these saliva samples appear to be like a very nice candidate for this alternative specimen collection for this virus detection. Next slide, please. So about this study a little bit. This possibility of rapid antigen test based on salivary diagnosis has received really increased attention over the past few months. So in our study, we designed this lateral flow diagnostic assay, this rapid antigen test, and is intended for identification of this SARS-CoV-2 antigen, and we used these de novo synthesized polyclonal antibodies to achieve it. And our study results represent the proof of concept that this rapid antigen diagnostic tests targeting this virus could be used in saliva samples of COVID-19 patients. Next slide, please. So, in the beginning of this study, we used, in collaboration with ATG service, we used this artificial antigen. It's a really unique one because it is made based on all 4 viral proteins. So it's like chimera. It's why we called it anti-4 epitope antigen. And so these antibodies were obtained. These antibodies were like polyclonal rabbit antibodies. So we have tested these polyclonal rabbit antibodies to test these abilities to create this test. Unfortunately, these results were not very encouraging. Nevertheless, we were able to build up this rapid antigen test. But as you can see, this positive signal from this anti-4 epitope antigen antibodies was much weaker than the competitor one. You can see by this red triangle, when we must see this positive signal, and in one case for these anti-rabbit antibodies, it's quite weak. So next slide, please. We had to switch to another target. In that case, we used this recombinant receptor binding domain called like RBD protein, which was kindly donated to us by Professor Kasper Tars. And in our study, we used this recombinant protein to immunize mice, and we collected quite many serum samples. So we used these serum samples and purified these mouse polyclonal antibodies and used these antibodies like a capture antibodies for this novel rapid antigen test. And now we achieved quite nice result. This positive signal was strong enough to proceed with this variant. Next slide, please. In parallel, we also had to create this in-house RT-PCR test, because in this situation we had to overcome some reagent shortages and reagent delivery delays, but we are very happy that we did it, because we used this method, like a reference method, in our study. And the detection limit of this RT-PCR method in our settings was 5 RNA copies per sample. And so by using this novelly developed PCR test method, we also tested this viral load in all saliva samples which were obtained from COVID-19 patients. Next slide, please. So the first set of saliva samples, what we used to test our new method, was saliva samples which were obtained from COVID-19 patients within the first 5 days after onset of symptoms, like really very start of the disease. And so both tests were able to detect this antigen test and RT-PCR test. These both tests were able to detect the virus in these samples. So 80% sensitivity was for both tests. Unfortunately, there were some differences in these results. Next slide, please. And next, we used a second set of these samples. These saliva samples were obtained from COVID patients which were with moderate and severe disease. And the mean day when these samples were obtained, it was 10 days after the onset of symptoms, or approximately 7 to 6 days after the first COVID positive test. Next slide, please. So in this set of samples, we were able to show that indeed this viral load decreased over the time during the infection, and this viral load nicely correlates with the time from onset of infection. So here we obtained this significance level. Next slide, please. And based on these results, on these RT-PCR test results, on average, we can tell that conversion to negative occurred approximately before 9 days after initial testing, or 12 days after the onset of symptoms. There we achieved these statistically significant results. So this test positivity correlates with the time between symptom onset and sample collection, and also the test positivity correlates with this time between the last positive test and sample collection day. Next slide, please. On the other hand, this rapid antigen test, it was positive on average in 32% of these saliva samples. And it's not surprisingly, because these samples were mostly collected within 6 to 16 days after onset of symptoms. But nevertheless, you can see these positive samples are dispersed through the whole time between the symptom onset and sample collection. So again, there's some discrepancies between PCR test results and antigen test results, but it could be explained by this methodology and this sample set per se. Next slide, please. So this it's why for this rapid antigen test we did not observe any statistically significant correlation between positive test result and viral load, or between this positive test result and time between symptom onset and sample collection. Next slide, please. So we were able to draw many conclusions from this study. First, and the most important one, it's like both the SARS virus antigen and RNA were detected in saliva samples of COVID-19 patients. The accuracy of both these tests depends on many different factors, and it excludes the time from onset of infection, the viral load in the specimen, the quality, processing mode of the specimen, and also it depends on all chemistries employed in these diagnostic assays. And the next conclusion, very important one, that this rapid antigen test could be useful for detecting these active COVID-19 infections at approximately 5 days from onset of infection. And also saliva this testing may increase testing capacity because the collection of these samples is much easier than swab samples. And so this implementation of saliva-based COVID tests can work like in point-of-care places like hospitals or clinics, or in some outbreak control spots like schools, airports, and nursing homes. But also we must stress out that these saliva samples must be optimized for different collection and processing variables and storage temperatures, transport and time to assay to determine best practice before we use these tests in routine. And also the last conclusion is that it seems that this RT-PCR test in saliva samples could be quite useful for viral load monitoring in hospitalized COVID-19 patients. Next slide, please. So it's an acknowledgement slide because very many people worked on this project. The special thanks to Agnia Kivran and Viktorija Gumnova. Thank you, because they both mainly designed and all these tests and also processed all these samples. And funding was received from this VPP project. Thank you for your attention.
Yes, thank you, Dr. Ranka, for this, I should say, very exciting story. And the way how we fail as researchers and find another new solutions. And I think Latvia is among the pioneer countries using saliva tests for mass screening. And it's not exception also for new antigen, rapid antigen tests. We are elaborating those tests exactly on specifically on saliva. And we participated also in discussion with a rapid response in the British Medical Journal about saliva as is it proper material for mass screening, and really we shared our experience here in Latvia, and I think we contributed a lot to development of this specific field of research. And of course Chinese manufacturers already are producing rapid antigen tests using our ideas partially, but really we promise to deliver on market a much more sensitive and specifically to saliva invented test. So now I want to turn to the third technology we developed in this national research program against COVID. And this is the project led by engineer Dr. Dmitris Bliznyuks, who is a leading senior researcher from the Riga Technical University, but he successfully joined the team of clinicians working at the Valsts Clinical University Hospital. So, Dmitri, the floor is yours.
Hello everyone, dear listeners and colleagues. I'm Dmitris Bliznyuks, and as Professor said, I will show you our results in the treatment part of our project. Next. So as you might probably know, please next slide. The COVID strikes exactly onto the lungs, and the pneumonia is the problem that we should face. Next one. As you probably have seen, there is a special devices, the huge ones, that are put inside our airways, and they cost a lot, but actually they are used only for the critical patient. Next one. But for low-risk patients, and actually the major part of the patients, much simpler device is used. So the price is still high. It's used by just attaching the mask to the patient. Nevertheless, the device is pretty critical because it helps person to breathe. So it puts additional pressure to the airways. And of course, it adds an oxygen to help patient to saturate the blood with oxygen. Okay, next slide. And as you see, as soon as pandemic struck, a lot of engineers started to work on their copies of these devices, so basically to breathe and to add oxygen. And as you might see, even the larger companies like NVIDIA, like MIT University, Rice University, and some else, designed a system that mostly work, I suppose. Nevertheless, they look a bit scary, at least for me. Next slide. So is there any other solution? Actually, yes. If we look a bit broader, there is a number of devices used in home care for so-called apnea therapy, and they do exactly what we need. So they provide additional positive pressure, so basically help person to breathe. They are low cost, so under 1,000, probably around 500 euros. And the most important, they are widely available. So basically, if these previous devices, they couldn't be manufactured so fast, that's why actually all the teams try to make copies to support the clinician. But this device is freely available. Of course, it's certified, and it does this main critical function to provide the additional pressure. Next slide. But actually, it's not providing the oxygen or provides it in unprecise way, because for this type of devices, it actually is not needed. Next slide. So the task would be like to add additional oxygen, to measure it precisely, and to connect it to this CPAP home care device. Sounds pretty easy, but if we try to add the oxygen, so on the left side we did some simulation. The red tube is the inlet of the oxygen, and it actually is not mixing itself in the tube. And if we try to measure it, then we receive a pretty large error, and of course it will be unusable. Okay, we can take some kind of a mixer. On the right side, you see the mixer. It will provide nice accuracy for measuring, but it will add additional hydrodynamical pressure to our system, and the treatment will not be as well. Next slide. So the problem is understood. Basically, we should design some kind of an add-on to this home care CPAP devices that would be capable to deliver the precise saturation of the oxygen, and of course, by providing this additional pressure. Why this precise is so needed? So for home care devices, actually the patient is stable. That's why he stays home. He could add some uncertain amount of oxygen or approximate amount of oxygen. As soon as the patient is in the clinics, his status could change very briefly, and to critical maybe as well. So the clinicians, they should know exactly how much oxygen is breathed in, and then they check by blood analysis. They understand how much, what part of the oxygen is really came to the blood, and that way they understand the therapy. They may change it, and that's why they should need precise oxygen saturation. Okay, so since we should test our device, we should receive allowance from the state agency, from the ethical committee, because we did our test on the real COVID patient. And device should be not just quickly or cheaply done, but it should be able to produced by mass. So the design should be mass productive. Next slide, please. So here is a slide with overall schematics. It's not so detailed because the project asks us to provide a patent, and since the patent is not yet published, the design is overall. So on the left side, we see a CPAP device, so this certified device that we use for the most critical function, and that is adding the additional pressure to the patient. And by supplying the external oxygen, the pure oxygen, we use a certified medical device to change the flow of the oxygen. And by using the buffer tank, we mix it and to provide a stable oxygen saturation. On the right side, clinicians use the standard tubes, the standard filters, masks, and so on. So basically, it's very easy for them to use it. And the device has a touchscreen interface with electronics to control. Next slide, please. And this buffer tank is really needed because it's not only important to measure it precisely, but as soon as the patient is breathing, so blue line shows us if you just provide a pure tube, so without any buffers, the concentration will change as the person breathes. So all the spikes are the breath cycles of the patient. But if we add this tank, and that's one of our ideas, the concentration stability is high enough into the almost at any breathing stage. Next slide. And here you can see the device itself. So it's on the right side, it's a photo. So black one is this existing certified CPAP device, and below the white one is our device. You also saw it in the video. So it's produced pretty easily because it's only cutting and bending operation on the metal parts. LCD screen can be used with gloves as well. And the tank inside is again reusable device. It's coming from other medical devices, and again we don't need to produce it, and it's already certified. Please, next slide. So we created our device. Of course, there was a number of iteration between we come to this final design. And in clinics, we validated our device against certified this high-cost non-invasive medical device. So the patients, actual COVID pneumonia patients, were tested for the first hour with this NIF therapy. And then with the same therapy, basically with the same pressure and oxygen, then they brought to our device. And again, for 1 hour, they were tested against our device. And each 15 minutes, the blood analysis were made to later on to analyze what was their status. Next slide, please. So here we see that overall, we tested 16 patients and 12 patients were left for the analysis for the different reasons. For example, the status of the patient could change very rapidly. And of course, the health of the patient is much more important than our test. And they just stopped the testing and changed the therapy. And the patients were with type 1 respiratory failure. Next slide. So let's look on the graph and plots and the results. So as you might know that pure air contains 21% of oxygen, and this therapy, it should raise, by using this therapy, we should raise the oxygen concentration in air along to positive pressure. And in some cases, some therapies allow to use even 100% of oxygen, but we choose the most stable patients which are not critical, so the result would be as accurate as possible. So our range was from 30 to 70% of added oxygen. Next slide, please. And what is the aim? So basically our aim and the device and the therapy aim is to provide enough oxygen into the hemoglobin. Probably you have heard about these devices, pulse oximeters that plugged on your finger. So they actually do that. They check how much oxygen is inside the hemoglobin in your blood, actually. So everything above 94% is okay, so that's the target. And on the left side, you can see the blue line is this reference, so high cost device, and the red one is our device. And you can see that in most cases, the lines are really close together, so the ranges is also compatible, but two patients with some different results could be seen. So let's look on them as well later on. So the next parameter, please, next slide, we checked in the blood with a blood analysis was so-called partial oxygen pressure, and there might be a situation where we actually deliver enough oxygen into the blood. So there is oxygen inside the blood, but hemoglobin is, due to some reason, to some diseases, is not absorbed into the hemoglobin. And again, here we see that device acts pretty nice. So all the lines are close to each other with some differences in this two situation. Next slide. And the last what we have, not the last that we have checked, so we have checked a lot more, but in this presentation, this will be the last test, not just to make it not so hard to understand. So one more parameter is gradient, alveolar arterial gradient of the oxygen. And again, so the graph you can see on the left side is the reference, on the right side it's our device, and the axis is not from zero, so they're specifically zoomed, and the results are statistically, the difference is statistically insignificant, so the device is actually as efficient as this SNF device. So talking about this specific patient, while testing them, that it was clearly seen that they have a hyperventilation, or basically they were breathing pretty fast. And since the first device is this reference device SNF, patient actually got along with this device, felt much better, started breathing slowly, and therefore our device is shown the smaller concentration. So basically, if we switch testing, if our device was first and then was this reference device, the situation with these two patients would be completely opposite. So that's why we can see that our device is really good in all the cases. So next slide. And by comparing the functions and the price, next slide, please, we can see that our device is on the left. Of course, the most important is the price, and not only the price, but the availability. So we can produce it pretty fast, and all the functions are still the same or pretty similar. So next slide. With that, we can conclude that our designed add-on device to this home care certified system is as efficient as this existing NIV device, and it could be used for treating the COVID-19 pneumonia, and not just that. Thank you for the attention. All the credits are below.
So thank you, Dr. Blisniuks, for this really the demonstration of very nice or also aesthetically principle and device itself. And this work package surprises me the most, actually. And I'm very satisfied that that team consisting the best engineers in Riga and the best clinicians at the lung disease department really were able to create the prototype and validate in very short time. And I think it was last week we submitted to the European Respiratory Journal, which is one of the highest impact journals in lung diseases, a research article on this device. So please follow the European Respiratory Journal, and we hope to get to publications as soon as possible. So now it's time to turn to discussion, and I'm very happy to give a word to Emil Sundjukovs, who is also part of our team. And Emil is a talented young information scientist or computing scientist. Emil?
Yes. Hi, everyone. Good afternoon, dear audience, peers, colleagues. I'm happy to see you here and our conference. And with me, I will have all of the three presenters from all of the three packages, work packages, or all of the 3T in our 3T project. So, so once again, just to elaborate to the audience, so we have an ability to also ask questions via the Slido tool. And please, I really urge you to do that and ask questions. So we'll be happy to elaborate on that. So yeah, so during the discussion, we will touch base basically in a simple or society or industry oriented language, what would be the fields of application, the unique selling points, and how our scientific groups are envisioning the next steps and the potential of the technologies that we're researching during the project. So dear colleagues, so the first question to all of you, and let's start our discussion with a question to all of the three colleagues of ours about the fields of application. So you have mentioned about the scientific background and also scientific advancements within the project, but basically in simple language or in everyday application. So where do you envision your technology in particular can be used to ease our everyday life and to ease the process of fighting COVID? Maybe let's start with Jānis, if you can hear me.
Okay.
Hi,
yeah.
Jānis.
Emil, thank you.
Yeah.
Yeah, I think for this my device, what I presented is relatively simple. This answer, yeah, we can use this in every room, yeah, especially in rooms where where many people are coming in. Where the situation is changing, yeah. So I would say more in public rooms, but also in offices, in different, as we have shown in hospital was the example. I think all these rooms are good places to apply our system, yeah.
So basically, with what you're saying, your technology might ease the process of getting back to schools, universities, and even offices.
Yeah, and maybe theaters.
Oh,
also,
sure.
Yeah.
The cultural events are very important, yeah, and this is what the society is speaking about. Thanks a lot for elaborating on this, Jānis. So maybe let's switch to our second T, which is the testing phase. And Renat, would you be so kind to elaborate on your vision on the fields of application, where we can place such technology to ease our everyday life?
Yeah, it's quite easy answer because we all want to go back to our normal lives. And to do that, we need to contain this virus. And the most importantly, we had to identify all these infectious persons as fast and as quickly and as efficiently as possible, just to protect other ones. So this test could be quite a nice advance in this testing, because it's fast, it's rapid antigen test, so this time is fast, and it could be used on quite simple this sample. The saliva samples, it's more easily obtained than these swabs.
So basically, with what you're saying, this is something in the future that we can buy in a drugstore, place it in the office, and use our saliva to test the coworkers or pupils, or
approximately like that, yes.
Okay, so I think it's a very wide application. So to ease the process. So in order to go further and switch to other questions as well, so I will also give a word to Dmitry. So on the same question. So what do you envision as a potential field of application for the respiratory apparatus you were working on?
Yeah, so our devices, it's again, the answer is pretty easy. It's not limited to COVID pneumonia. Basically, it's not even the pneumonia, actually to all so-called type 1 respiratory diseases. So that's very easy and very broad range where device could be used. And actually, if this system would be produced in as a masses, like thousands of devices, after this pandemic ends, it would be could be reused for the sleep apnea testing, curing, because even in Latvia there are around 20,000 patients with apnea.
Okay, so with what you're saying is also that we can not only use this in the emergency situation, but basically we can pass it along to people of need in other disease domains where we should have oxygen-rich environments.
Yeah.
Perfect. So also coming back to the Slido and the question from the audience, and I will try to elaborate this on the next question about the unique selling points. So each one of you, dear scientists, dear colleagues, you stated that for sure there is a state of art and there are universities or even companies like Dmitry's elaborated. So one of the automotive concerns is also looking into designing the machinery for respiration. What would be the unique selling point for the stakeholders and in comparison to the state of art? Maybe we can start with Renata that time. So there is a popular, I think every one of us at least once tried out the nasopharyngeal swab, which is sometimes, a painful activity, but nevertheless, this was one of the state-of-art principles how we can get tested for COVID. So what do you see as unique selling points for your approach? And is it actually as precise as the one that we used to have?
Well, about this precisity, saliva is very complex sample because it can vary. It can be variable. So it depends when you take this sample in the morning or after a meal or after or before toothbrush. So before we are going forward with this test, we definitely need to understand how to collect these samples and in which time point this result will be the most positive, maybe like that. So it's very important. Yes, it's easy to collect, but it's a little bit more complicated to validate in comparison with this.
So but basically, but by using the right instructions, we have on the table the cheaper, faster.
Hopefully, yes.
Okay.
Yes. And yeah, another plus is that hopefully we do not need like attendance from this medical personnel. So person can do it by it or herself. So it's the plus. Also, yes, it depends.
So basically what you're saying is quite hard to do the nasopharyngeal swab test by yourself. So I've seen a couple of people just touch base in the nose.
It is quite hard, yes. And in the literature, there are some results that, yes, self-testing is not as accurate and yes, much less sensitive than which was conducted by medical personnel. It's obvious.
So with saliva is a potential way to go. Thanks a lot, Renata. So switching to Yanis. So if I will turn on my phone, at home I have my own home-based sensor measuring CO2, temperature, humidity, and maybe small particles even, just to see the quality of the air. So in comparison to just regular home-based sensors, what would be the value proposition of your technology and this so-called selling point to the stakeholders?
Yeah, I would say I'm a little bit wondering why there are no such devices appearing in the world. Yeah, I have not seen. Yeah, what is well accepted is really that people are understanding that this system of measurement of CO2 concentration is important, and everybody knows you should reduce the CO2, and then it's good. Yeah, but our system will show a lot of addition. Will show more important points. This coughing and sneezing events in the room. Yeah, that's really much important than the CO2 concentration. Therefore, of course, you can imagine also to develop this system easier, maybe on some mobile phone application. Yeah, also would be possible for this sound recognition. Yeah. But I would say that our system is much more precise, yeah, and it will cost only 2 or 3 times more than system with only CO2 and temperature sensors, yeah, and the advantages will be or the precision will be orders higher than these systems.
Okay.
Also. So.
Thanks a lot, Yanis. So I cannot, just pass this question also coming from an audience and partially for myself. So for sure, we're speaking about also sound monitoring and sound classification So does your technology also allow privacy-preserving sound qualification? So we don't, not overpassing any, privacy challenges here.
Yeah, this is a bit, of course, negative issue of this system of recognition. Yeah, at the moment we are working in following way. We are sending the images to our server and analyzing there. Yeah, but the sound is analyzed on the spot. It means in the room, but this will cost a bit more. We have some this NVIDIA small computer there installed. It costs additional €100. I think it's normally, as with other systems, as video surveillance systems, there are rules and there are prescriptions how to do this. Yeah, and we also use Google Home and so on. Yeah, that's it's a point, but which can be solved somehow. Yeah, but this but we cannot do image analysis in this sensor system. It will cost too much. Yeah, so we need a server for this.
Sure. So with what you're saying, I think it's important to elaborate that it is also possible in industrial wise to do calculus on the node or on the machine itself. So and provide such kind of deployments, which giving us quite a good benefit. Speaking of privacy preservance. Thank you so much, Yanis. So Dimitris, you mentioned in your presentation that there are a lot of institutions who tried, to hack and to adapt the existing systems or create a new one towards helping the patients to ease the process of treatment. So what would be the unique selling points in your case, apart from the cost that you elaborate in the presentation for sure?
Yeah, of course, the cost and the most important ability to produce it. As for uniqueness, as we can see, the most of the projects, they try to copy basically all the system, even this critical part, and I suppose they will fail or it will be really hard to get the certification. Our part is just adding the small step. And there was similar project as well. So this Mercedes device or F1 device, it's also pretty simple. So it reuses other components, but again, it's not so precise in the terms of oxygen measuring. So ours is like basically in the middle. It's not trying to copy all function and is not so simple that it's not unreliable and unprecise.
That's amazing, Dmitry. So what you're saying that we're more precise than Mercedes, and I'm waiting for the first car to be produced by your group so we can beat up.
Yeah, they had a bit different aims. So our aim was to help clinicians and actually to detect the treatment.
Sure, and passing along. So every one of you mentioned. So you have your own domain of expertise. So both computer science. Mathematics and modeling, and also the biotech or the biology background, medical background. So what was the most, so in the process of interdisciplinary approach, actually tackling with your knowledge and tackling the problem where your own competence is not enough, you need to co-work together. What was the most exciting part or lesson learned during, working on this project? Maybe Dmitris, you can start with that.
Actually, it was very interesting and fun, I would say, to work in such a multidisciplinary team. I really thank my colleagues. It's really interesting persons, and for me, if I should point one, quite a challenge and quite a new feelings was when I entered this COVID area. That was something different when you enter this suit and you go inside and check how your device is working. That's something different.
This is something to remember for a long time. So thanks a lot, Dmitris. What about Renate? Do you have anything to tell us on that?
Yes, probably it was like a turning point when we started to understand that the first solution will not work as good as we wished for. And so we find this possibility of collaboration with another project, and we have got this protein to work on. So probably, yes, this one, this collaboration showed like a Excellent. These ideas, to explain these ideas and to communicate, and so to know what what another project is producing, and we can just switch these ideas to work on.
That's amazing. I'm really amazed how a scientific group, sometimes very conservative, can adapt quickly, like you yourself. This is kind of a startup mindset that you were using during the project. Jānis, what about you? So you're coming from a mathematical modeling field. So how it was on your end?
Yeah, of course, in the mathematical modeling, we are dealing very much with the modeling of different physical systems, of different technologies. I have worked really on a wide field. Yeah, but anyway, this was very interesting project. The first, we worked together with our colleagues in the Faculty of Computer Science with Leo Silava, Guntis Arnitsāns. We learned also a lot of how the system should be designed with the data transfer and so on. Yeah, and the second was, of course, that I also learned a lot of medicine a bit, yeah, in viruses here, and of course we did a lot of studies to build this model. We needed really to find the best data from literature, yeah, and we were also very surprised that there is no data for distribution, how the droplets are expelled, yeah, the size distribution, and the best data was from 1946. Yeah, so it was really very interesting experience. Yeah.
Interesting. So the follow-up question to Johannes actually also coming from the audience. So we have faced many different strains now, and it will also be the question to Renat afterwards. So many different strains coming from Africa, UK, other regions. So how adaptable is the calculation system that you designed? So can we use it for potential new strains? Hope it's not going to happen, but nevertheless, yeah.
Yeah. Probably some new strains will happen with us. Yes. So unfortunately, I think, yes, yeah, it is adaptable. We are using now this number, what I said, that we, from last year, from this original virus, yeah, that we need approximately 300 virions to absorb to get infected with a 50% probability. And if we know these numbers for another strains, we can put this just in the model, another number, not 300, but different numbers, so that we get a higher probability, 60% higher as for this UK virus. Yeah, so and then mix these probabilities. That's so easy.
Mm-hmm. So an admin panel is there to change the variance. Very cool. Renate, so the same question, I think it's very much applicable to the testing perspective. So what are your thoughts on that?
Well, if we are talking about these mutations, it's really not good that these mutations appeared so quickly, and it definitely could impact these RT-PCR results, because this method is quite sensitive to all these changes in genome. Otherwise, for this protein point of view, we are using these polyclonal antibodies. So these tests could be less sensitive to these mutations, because we are talking about proteins, and these point mutations will not change, could not change these proteins dramatically. Of course, we do not know how it will go further in future, but definitely we have to stay agile about these new variants. But it seems that, yes, this rapid tests will suffer less a little bit.
Thank you so much. So switching to Dmitri's, not about the strains, more about the engineering perspective. So you mentioned that it will be, how to compare. So basically an add-on to existing device, so I can plug and play. So there is a term like that, easing the process. So how easy it is actually to adapt. Imagine, I don't know, we would have here 10 machines as you showed us, and how easy it is to attach your your know-how to
Yeah, basically we would say it's like a plug and treat system. Since we're using the standard connections, we actually use the right tubes from the clinician. So we didn't bought ours. We just asked for the clinician. They gave their tubes and they perfectly fit. So it's very easy.
Okay, so plug and treat. Let's remember this slogan. Very interesting. So a question to Renata coming from the audience. Is any additional testing required to confirm the test result?
At this moment, this World Health Organization recommends that additional testing is required when you have this positive test. And that also depends definitely on this epidemiological situation, because when we have a lot of these COVID positive cases and your result is positive, so it's definitely it's a big probability that you have this disease. And otherwise, in this different settings, so we can question these results more in the more details. So yes, at the present moment, this World Health Organization recommends this retesting.
Thank you so much. So just once again to following up on another question. So this test can be adapted for the home use. There is another question coming from the audience.
For what?
Your test can be adapted for the home use, so it's not being sent to laboratory.
It can be adapted for home use. Yes, definitely.
Thank you so much. So a question to Jānis. So you mentioned about sensing the room or multiple rooms on sounds, images, other variables that are important to understand the quality and the risk measurements. So, but what about the everyday situation? You mentioned theater or opera. I think a lot of people would like to go to theater in the nearest future, hopefully. So how would it work? Would you envision any application like mobile app sensing and stating, hey, you better stay home, or you better, I don't know, open the window now in the pause? How you envision this communication with the individuals in the room?
Our proposal was to install together with this system, maybe in the same box, some signals with a red light or green light or yellow light. It would be the simplest situation, and right then it's safe or not safe. Yeah. Second possibility would be also to add some app when you are going in the room, for example, in some theater or to school. Yeah, you have in the school some server central which gives this information to mobile phone. Yeah, I can imagine. Another possibility, of course, is also to use this model to predict some potential situations, not knowing exactly how many people are in the room. You can give for the model, for example, I have my room 100 to 50 meters size, and the ventilation is so and so, and there would be so much people, yeah, and the probability of infection, this prevalence of a virus in the community is also known for the real time, and then you can calculate in how long time the probability of infection will grow. So I can imagine such 3 scenarios maybe.
So it sounds like a very good added value for the businesses to predict before even people are coming.
Yeah, to predict with this model and to say, okay, we cannot put 50 person inside here. Yeah. There are, of course, some regulations, yeah, already known how many people can be in one room and so on, but we can maybe predict much more better these models, these numbers. Yeah.
Sure, perfect. So in order to summarize, and then also have a final question to Professor Pirax as a leader of the whole project. The question is the following. So I hope that both our live stream and also the recorded video will be spread, and there will be interest coming from different stakeholders. So the 2 small questions to each one of us, each one of you, is who you're looking for And what's your group's next step towards bringing this scientific project to the market? Maybe let's start with Dmitris. And
yeah, actually, we might be interested in additional testing because, as you saw, it's only 12 patients. The project is over, so device is ready, but we would be also happy to test it a bit more. So some additional help will be needed from funding, of course, and the clinician side as well. And we're already taking the steps to make the device even better. And actually, it's also connected with the question about this home use. We already received one European project that will actually look on this home use of the similar devices.
Great, very interesting. Renate, what about you?
So definitely we need to expand this testing because we need to look when and under what circumstances this test will work the best. So we need to obtain somehow this bigger cohort of patients to involve in this testing and just to get more data to analyze. So of course, another step forward will be to make this device a little bit cheaper because we used this commercial basis and it's not really cheap. So, but we need to look also to for a little bit modifications.
Thank you so much. Jānis, how about your first or T1 project?
So what we think we should do still, yeah, I think one point is to increase the precision of measurement of number of people. It means train the neural networks more, yeah, and look on more different situations. We have tested this on relatively small rooms. We tested also with 2 cameras for larger rooms, yeah, but this is a point where we can, I think, improve still the precision. And what we are looking for, yeah, I think some company who has experience in electronics and producing of sensor systems, yeah, but also with a background with this network communication, yeah, we need some systems, this cloud system, where this management of the whole, I would say, community, because each room, which is separate, yeah, will use some global computing center, I would say so. Yeah, therefore some electronic industry. Yeah, in Latvia, I know there are companies producing sensors. Aranet is the name everybody knows. Yeah, so but yeah, another of course also.
Thank you so much. So in the place of summarizing our discussion, I will pass a quick question to Professor Piraks, who was throughout the whole project and also today is holding an umbrella above all 3 T's. So Professor, if you're with us, what would be your call to action to the ones watching our live stream or recorded video? So who are we looking to, and what will be the next step?
Yeah, it was very important for us to organize such a virtual conference, or we were actually calling it a Jahrmarkt. In Russian, it's Jarmarka. In Latvian, Gada tirgus. That means peasants are selling their products to customers. Of course, in the old times, it was quite easy to contact directly. Currently, we need internet, such international internet platforms to find proper investors. And of course, we are interested in investment, in private investment. What we will do the next, we already started the patenting, so the intellectual property rights will be properly fixed and solved. So, and we are forward looking to the investors, and we will try definitely avoid somebody who will just try just to adopt our ideas and produce something which is probably of less quality. So we are looking for serious investors, and you see that our products are really on a high stage of development, so we are ready to do it.
Thank you so much, Professor. So thank you, dear peers, dear colleagues, for this short but I think fruitful panel discussion. And prior to giving the words further on, so what Professor said, we're looking for collaboration partners, and this collaboration is not happening only without the fruitful ground, which is being given by Investment and Development Agency of Latvia. And I'll be happy to give a word to Anna Janberga, who is a leading expert technology scout at Investment and Development Agency of Latvia, who will give us very practical how-tos. If you're the industrial or clinical or any other partner willing to collaborate with the scientific groups, how you can do that and how you can achieve this collaboration. Thank you so much, Anna. The floor is yours.
Hello, everyone. Good afternoon. And thank you, the organizers, for the nice event, and also for giving me the opportunity to present how we at the agency can help our companies and research organizations. Please, next slide. One of the approaches we are currently working with at our agency is the development of ecosystems in our defined five smart specialization areas. And of course, one of these areas is biomedicine, but we are also, as you know, we are having the smart specialization areas in smart materials, in smart energy, and ICT. So our aim is to work with all these smart specialization areas and increase the value of these areas. Please, next slide. And currently at LIA, we are working with three ecosystems. So one is in biomedicine, one is in photonics and smart materials, and one is in smart cities and smart mobility. But together with OECD, we want to have at least one ecosystem in each of these five smart specialization areas. So together with OECD, we will do the identification and of knowledge-intensive value chains in Latvia, and also we will map out the ecosystems. And the goal of the ecosystems is to bring expertise from industry, from researchers, from public sector, so we can define common goals and challenges in these smart specialization areas, and we can achieve increased values of our economy. And the role of LIA is to bring together these ecosystem members and also to proactively facilitate the goals that we have defined for our research and innovation smart specialization areas. Next slide, please. And currently, our biomedicine ecosystem revolves around medical data lake and medical data and research data usage for research and innovation activities. And in our ecosystem, we have members from companies. So we have from our local companies, and we have also from the international companies. And of course, we have also researchers. And in this ecosystem, we have also particularly representatives of largest hospitals. And also we have representatives of ministries. So together we can achieve more and develop this area better than just fighting alone. Next slide, please. So one of the practical ways how we can help our companies and research organizations is together with LIA representative offices, we can help you find partners for research and innovation activities, or also in this case, maybe for licensing purpose. And we currently have 11 offices in geographical Europe. We have also one representative in USA, and we have representatives in 4 countries in Asia. And our representatives can provide services not only exclusively to our ecosystem members, but also to other Latvia's companies. But for the other companies, it's for charge. And our representatives not only can help you with helping with making dialogue with possible partners, companies in other countries, but for the companies, they can also provide services related to knowledge of the local market and just to give sense what you need to be able to reach this export market. Next slide, please. So from the 1st of April, we will have also one new office in Brussels in Belgium. And the main tasks for this office will be to promote Latvia as a country with knowledge-based economy, so that people who are working in European Union structures, they know that Latvia can be a useful partner in different type of activities revolving around research and innovation activities. Secondly, the aim is to create new collaboration opportunities for our companies and for our research organizations. For example, looking for partners in Horizon Europe projects. As we know that in this new period, the Europe is having this. You really need to cooperate between different stakeholders, and we know previously researchers were maybe working more with researchers, so they really know what is happening in research environments. But more and more, European Union wants that researchers are collaborating together with companies. And the third goal is to inform our ecosystem and our companies about different EU programs and initiatives. Of course, if you're talking about more research traditional programs, it will be under VIA and LZP, but for companies, LIA will taking the main role in this part. And of course, as we know, a lot of information is shared in informal conversations. So it's very difficult sometimes to get into these already closed needs of consortia. But in this way, maybe we can open new doors and get into these already formed consortia in Europe. Next slide, please. And in this slide is just short overview about different incentives and support mechanisms which we have at our agency. Mostly these incentives and support mechanisms are made for companies, SMEs. Some are also for business idea authors, and some of them can also apply to researchers who want to know more about innovation activities. Next slide, please. And in this presentation, I will just briefly touch on two of our funding mechanisms for development of new products and technologies. The first one is Innovation Voucher Program, and it's for SMEs and also large companies. And we have currently 4 types of innovation voucher. And for example, in the Get to Know Me voucher and Classic voucher, the main reason why they are made is that we increase the cooperation between our companies and researchers. So companies can develop their product together with research organizations, and LEA is funding the researchers' work. And the funding is up to 25,000 euros from LEA side, and this program is currently open, and this 25,000 euros is for one idea. So that is maximum amount for one idea. Next slide, please. And the second program, which will be soon opening, is together with Norway, so shortly called Norway Grants. There will be 4 activities, but I will also just give you brief introduction to the small grant schemes. So the small grant schemes is also meant for development of new products and technologies. And for Norway grants, it can be obtained. So it's for the SMEs, only for SMEs. The development of green innovations and ICT products, this call will be open starting from the 2nd quarter this year. And the call for welfare technologies will be starting for 3rd quarter of this year. In this program, the maximal funding in small grant schemes from LEA will be up to 100,000, 30,000 of euros. And maybe what I wanted to also add about innovation voucher program